So our last concept in our cells unit is about the cell cycle and how that gets messed up whenever we are talking about cancer. So everything always starts with a single cell. Once the sperm fertilizes an egg, that fertilized egg is called a zygote. And so that single cell is going to go through cell division repeatedly. So once I have this grouping of cells, mitosis is going to continue to happen to give rise to many identical cells. So now I have this group of identical cells. However, that's not going to be forming a body. So differentiation has to happen. So you hear in that the word different. So that is when those cells that creates specialized cells that have specialized structures and functions. So specialized cells then become tissues, those tissues become organs and develop organ systems. So that's why in fetus development you have certain milestones where you have development of brains and hearts and other organs because those cells have to differentiate. So whenever I'm talking about cell differentiation, before that time, those are called stem cells. So these are cells that are undifferentiated, and that means that they can become any different type of cell, any different type of differentiated cell that's specialized. There's two types of stem cells. There's embryonic stem cells that are cells that have never differentiated before. So that is going to be early on in fetal development. And then you have adult stem cells. So these are cells that are specifically found in your bone marrow, and they are partially differentiated. So what that means is that they can't become any type of cell, but they can become bone, blood, cartilage, fat, and different connective tissues. So they can become a wide range of types of cells. So just a quick reminder, whenever I'm talking about cell differentiation, I'm talking about complexity of the organisms themselves, of these multicellular organisms, and that goes back to our levels of organization. So we know that cells are the basic unit of life that has all the characteristics of life, but then when those cells group together that have the same function, it's going to make a tissue, so a group of cells working together for a common function. When those tissues work together for a common function, that's going to be an organ. When the set of organs that work together for a common function, that's going to be your organ systems. And then whenever you have all organ systems that are working together so that an individual member of a species can survive, that makes the organism. And that leads to cells then moving into the cell cycle. So the cell cycle is a repeated pattern of growth that's going to include DNA duplication and cell division that's going to occur in eukaryotic cells. Because remember back to our difference between prokaryotic cells and eukaryotic cells. Eukaryotic cells do mitosis in order to divide. There's two purposes for the cell cycle in mitosis, and that is to grow and to repair damage. So remember, growth doesn't happen because your cells get bigger. Growth happens because your body makes more cells. So when you went through puberty, your body created millions of more cells to make the inches of your body. And also when you damage your cells will complete mitosis and duplicate in order to replace the damaged ones. So the cell cycle in general has three main phases. The first phase is interphase, and that's whenever the cell's going to grow and do its job. The second part is mitosis, and that is specifically when your cell is dividing. And then the last part that kind of tags along with mitosis is called cytokinesis, and this is whenever the cytoplasm of your cells officially splits and creates two completely separated cells. So here is a visual representation of the cell cycle. You have, on the very outside this circle, you have interphase and then the mitotic phase. So we're going to go into the specific phases of mitosis: prophase, metaphase, anaphase, telophase, and then you have cytokinesis in there because that always immediately follows mitosis. And then you have specific kind of subparts of interphase. So speaking of those subparts of interphase, we know that interphase is whenever my cell's going to grow and it's going to be doing its job. And that's broken down into these three parts, G1, S, and G2. So G1 stands for gap 1 phase, and this is whenever the cell is going to be growing and making proteins in order to function and do its job. S phase stands for synthesis. To synthesize something means to create something or make something. In this case, we're making DNA. So DNA is going to be replicating, and that's going to double the number of chromosomes. And then in G2 phase, that stands for gap 2 phase. And this is when I'm going to do the same thing that I did in G1. The cell is just going to continue to grow and continue to make proteins and do its job. So it's important to note that at the end of interphase, the cell has 2 full sets of chromosomes. Because after I split, one set's going to go to one cell and the other set's going to go to the other cell. So really quickly, let's just go over some DNA vocabulary. We're going to be talking about DNA in different formats. So just a reminder, a chromosome is one long continuous thread of DNA that consists of thousands of genes and regulatory information. So think about DNA as a strand of yarn. Whenever it's tightened up and wound up into a yarn ball, that ball is a chromosome, where the yarn itself is still DNA. A gene is just a section of DNA that contains the instructions for making a protein. So think about multicolored yarn and how there's sections of blue and sections of red and sections of pink. Those would be the genes on the strand of DNA on that chromosome. So each human body cell has all of your DNA, and it's organized into 46 chromosomes. In normal body cells, chromosomes always occur in pairs of homologous chromosomes. We'll get back to the term homologous chromosomes later, but for now, moving on. So chromosome vocabulary. If we are looking at a chromosome, a chromatid is one half of a duplicated chromosome. So in this picture here, you see an X. That X is a chromosome that has went through S phase, and so the DNA has been duplicated. So I now have these 2 copies of the chromosome that are attached to each other. So half of that is a chromatid. When they are together and they are identical, they are sisters. So sister chromatids is the X, because I have duplicated chromosomes. Centromere, you hear center in that. That is the region of the chromosomes that looks pinched. It's in the middle. It's the center. And then the telomere is the ends. Okay, so the ends of the DNA molecule. So the tips of the X's. When we're talking about homologous chromosomes, you can see in the picture here, homologous chromosomes are chromosomes that you get one from your mom and one from your dad. So they contain the same genes on them. When we're talking about mitosis, we are done with interphase and we're moving to mitosis. Mitosis is just the division phase of the cell cycle. So it's when one cell becomes 2 identical cells. We call those daughter cells because they are like made from the original. So it's kind of like the parent cell and the offspring cells. There are prophase, metaphase, anaphase, and telophase in mitosis. So this picture is giving you a quick overview of what we are going to go through. So the first step is prophase. In prophase, my chromosomes are going to condense. So my yarn, my chromatin, that is the yarn, wraps up into the yarn balls, the chromosomes, and they're visible as sister chromatids. So remember, sister chromatids are when they look like X's. The nuclear membrane disappears. So in the picture, my nuclear membrane is still here. However, the surrounding of the nucleus, which is this black line here, is going to dissolve and go away. And then the spindle fibers are going to form out of the centrioles. So these little circles here are the centrioles. If you remember, those are the organelles that help with cell division. Here we are in cell division, so we're going to talk about them. And the spindle fibers are just these strands that are growing out of them. So it looks like the spider legs of the spider here. Next, we have metaphase. So we have spindle fibers connecting to the centromeres. Remember, the centromeres are the center, the middle of the sister chromatids, and that these sister chromatids are lining up in the middle of the cell. So think middle, metaphase. The next phase is anaphase. So A for away, anaphase. What that means, the sister chromatids get separated. They get pulled apart from each other by the spindle fibers. So the chromatids, they're no longer sisters because they've got broken apart. The chromatids are moving to opposite ends of the cell. So they're literally like being drawn and quartered. And then finally we have telophase. Sometimes you might hear somebody refer to this as telophase. It's the same thing. So this is when my chromosomes decondense. So they're still in chromatid form here. They're still wrapped up into the tight yarn ball, but that yarn ball is going to start unraveling back into chromatin. And my nuclear membrane is reforming. So it has reformed in this picture around the chromosomes to create two separate nuclei. My spindle fibers are starting to break down and go away, and cytokinesis is starting. So remember, cytokinesis is when my cell is going to split into two. I have this little divot here where I can see that the cells are starting to split apart. So when we're talking about cytokinesis starting, let's talk about what it looks like. This is the step where the cytoplasm officially divides into two individual cells. So here in my picture, I have two cells that are distinctly individual from one another. They have split. In plant cells, a cell plate is going to form between the nuclei. Remember, plant cells have a cell wall, so the outside of their cells is very rigid, and so it can't pinch in the middle. However, in animal cells, this cleavage furrow, this little dip here, can form because the outside of animal cells is just a flexible membrane. So in animal cells, that cleavage furrow forms and it eventually pinches so far in the middle to where one side of the cell touches the other side of the cell and then splits. So why does cell division need to happen, and how often does it happen? Well, every cell divides at a different rate based on its need. So for example, the internal lining of your intestines normally replicate about every 5 days. Think about it, your stomach acid is moving through them. They get damaged pretty easily. Your skin cells, about every 2 weeks. People are constantly rubbing into things, getting cuts, etc. So they need to replicate pretty quickly. Red blood cells go through mitosis about every 4 months. That's why there's a limit on how often you can donate blood, because you have to wait for your body to do mitosis of your red blood cells to make back what you donated. And your liver cells replicate about once a year. So why do your cells divide? Well, like we said earlier, it is so that your body can grow, or so that your body can repair itself, replace damaged cells in your body. So cells just can't get bigger and grow. They have to stay small so that they maintain a high surface area to volume ratio, because that's how they're efficient with going through cell processes and chemical reactions. So they can't just get big. They have to stay small. So in order for you as an organism to get bigger, you have to make more cells. With cell regulation, how do cells know when to divide and when not to divide? And what happens if that regulation fails? So your cell cycle is controlled by a chemical control system that starts and stops in the cell. And proteins are typically what's going to be these controls. Regulation is either external or internal. External meaning that the signals are coming from outside of the cell. So for example, hormones, nutrients, etc. So when you went through puberty, your body released hormones and your cells got these signals that said, all right, it's time to reproduce and continue to go through mitosis. We're going through a growth spurt. If your body has a lack of nutrients, if I don't have nutrients, then it's going to stunt my growth, meaning that your cells recognize there's not enough nutrients to go through mitosis to power your body to do that. And then you have internal signals. So that's signals that come from inside the cell, specifically inside the cell's nucleus. So this is when your DNA that's within the nucleus is providing the instructions for the cell and giving the cell the okay to move forward with replicating or saying to stop replicating. So within the cell cycle, there are checkpoints, and these checkpoints happen during the G2 phase, during mitosis, and at the end of G1. And this is when the cell just double checks that everything is okay, and either says, hey, stop, we need to fix XYZ, or everything's good, go ahead and move forward. When we were talking about lysosomes, we talked about apoptosis, and I said that we would come back and talk about why that was important, or why you would want your cells to kind of dissolve themselves. So here we go. Apoptosis is programmed cell death. So this is whenever either internal or external signals are going to activate specific genes that produce the self-destructive enzymes. So remember, those lysosomes, those organelles, contain enzymes, and what happens is they just like pop open and the enzymes release within the cell and the cell dissolves. So an example of why this is important, or a time where this happens, is during fetal development. Your toes and your fingers are connected in utero, and the cells that are in between your fingers and toes will undergo apoptosis so that you do not have webbed fingers and webbed toes. So in the picture here, what's happened is these cells right here should have undergone apoptosis, however they did not. So this person has webbed toes. And issues with cell regulation can lead to cancer. So cancer is uncontrolled cell division. And this happens when the regulation of the cell cycle breaks down, meaning that cancer cells divide much more often than healthy cells do. And this is going to lead to the formation of tumors. So tumors are just clumps of cells that are dividing uncontrollably. So instead of hitting the checkpoint and saying, hey, we have too many cells, we need to go ahead and stop and just stay in interphase instead of going through mitosis, it's just like, no, we're going to go through mitosis over and over and over again. So that's where that uncontrollable replication is occurring. When we're talking about tumors, there's two different types of tumors. There are benign tumors and malignant tumors. Benign tumors typically remain clustered together. A lot of times they are typically more harmless than malignant tumors, and they can be more easily removed because they are located in one place. However, malignant tumors are cancer cells that break away from the original tumor and can spread to other parts of the body. This is called metastasizing. So if a tumor has metastasized, it means that it has spread to one organ from another organ. So if you've ever heard of somebody who has a tumor in their liver, but they say they have lung cancer, it means that the original tumor started in the lung, and then that tumor was malignant and metastasized and broke apart, moved to the liver, and grew there. So a lot of times malignant tumors are the ones where you hear like stage 3, stage 4, and people have multiple tumors within their body. So what causes cancer? There's a few different things. You have biological factors like age. The more that your body has had to do mitosis, the more likely it is to make mistakes. Inherited genetic mutations. There are some types of cancers that run in families. For example, there's specific types of breast cancer that runs in families. That's why they always ask you for your family medical history, so they can kind of keep an eye on that. Skin type. We'll get to that in a second. Lifestyle choices. So your diet, your physical activity, your exposure to UV radiation, and your exposure to other carcinogens. Viruses and other infections. There is HPV, which stands for human papillomavirus, and that can cause cervical cancer. The cool thing about viral causes of cancer is that those are typically going to be more preventable because we can create vaccines to prevent you from being infected with that virus. So, cervical cancer via the HPV virus can actually be prevented with vaccines like Gardasil. So, that's great technology moving forward. And whenever I was talking about lifestyle choices, exposure to UV radiation, anything that is known to be a cancer causing agent is called a carcinogen. So these are specific chemicals that cause cancer by mutating your DNA. So some examples of those things are tobacco smoke and asbestos. So tobacco smoke is very well known to cause lung cancer. Asbestos causes mesothelioma, which is a type of lung cancer. So when we were talking about lifestyle choices, exposing yourself to specific carcinogens can increase your chance of cancer. So all of these things kind of have to do with increasing the amount of mitosis that your body has to go through. If you're damaging your cells more often than what they need to be damaged or have to be damaged, then you are increasing the likelihood of your cells accidentally making a mistake during mitosis and causing cancer.